All right, so I'll just make a short introduction. So Ulf is the CEO of NBE Therapeutics. It's a company that works with antibody drug conjugates, or ADCs for short. So he will tell us more in a minute, but I will just make a very short introduction to what ADCs are. Or do you want to make it?
No, it's great. If you make it, then I can learn something.
Just
That's
to make
fantastic.
sure it's short, you know? So yeah, they're basically antibodies attached to a toxin, so the antibody finds its target, so in our case, a cancer cell, releases the toxin and kills the cancer cell. So, I mean, in theory, it sounds really good, but obviously, so ADCs have been around for a while, and I know some of them have had issues, you know, with efficacy, side effects. So, yeah, I just want to ask you more about these issues. What are you doing at your company to try to fix these problems?
Well, I can talk for half an hour about this at least, maybe even for 2 hours. Yeah, exactly. Maybe I should first say, I mean, I am not prepared at all. So nothing of this is somehow rehearsed. It's totally spontaneous. So I don't even know what questions will be asked to myself. But yeah, if it comes to the technical thing and what are the difficulties with antibody drug conjugates, I mean, we find these molecules fascinating because the concept is very easy. So we attach something that is very cytotoxic to an antibody that is specific for a cancer cell. And as you said, that is a very easy concept that I can even explain to my grandma, and she will understand it. But the difficulties really lie in the design of the molecules, because you can imagine if the toxic moiety of these ADCs falls off after injection into patients too early, then it's not better than standard chemotherapy. So one has to make sure that one really has a good control about the integrity of these molecules and making sure that the toxin is really delivered to the site of the tumor or the tumor cell. And that sounds easy in concept, but it's very complicated.
Okay, so can you tell us a bit more on what specifically you can do to make this technology actually work?
Yeah, I will try to keep it very short. So I think the strength of our technology is, and I think that sets us a little bit apart from some or most of our competitors, is that we have our own patent-protected site-specific conjugation technology, which means we can precisely control where the toxin is coupled to the antibody, and we do this in a fashion that we have very stable bonds. So that exactly this problem that I outlined at the beginning is not happening, so that the toxin is not falling off prematurely and causes the standard chemotherapy side effects that you know from the clinics when you use normal drugs. So the site-selective or site-specific conjugation is key, but also the toxin we are using has actually the surprising feature that it not only kills the cells, but it also strongly activates the immune system against the tumor cells. So in a way, we have actually two mode of actions as part of our molecules, which we believe will be very important later on for a therapeutic success in patients. So not only killing the tumor cells, but also activating the immune system specifically against the targeted tumor. And we have great experimental evidence in, let's say, tumor models. We still have to show it in patients.
Okay. And I also wanted to ask you something because, as I mentioned, so you are developing a new generation of ADCs, but you know the technology ADCs has been around for a while already. So I guess my question is, why do you think it's better to develop a new generation of ADC instead of focusing on some of these newer therapies that are gaining so much attention, like CAR T therapies or checkpoint inhibitors or maybe even CRISPR?
Yeah. Well, I guess CAR T cells, immune checkpoint inhibitors are kind of, the molecules of this century or kind of this decade. And everyone knows about immuno-oncology, which actually was different a couple of years ago. I think 6-7 years ago, people were very skeptical about immuno-oncology and kind of leveraging the immune system to fight cancer. Of course, those are all fantastic principles. I think they are, as important for future cancer therapy and also current cancer therapy. And I think unless you have been in coma in the last couple of years, you know that CAR T cells are extremely successful. They can actually deliver total cures of cancer, which is actually quite a paradigm shift, because for many years, I mean, with even more targeted chemotherapy drugs, very often you just have a life prolongation, and it's not, let's say in very rare cases, it's curative. So all of a sudden, as molecules are developed or cells are developed that engage the immune system, we have all of a sudden the possibility to really cure cancer, and that gets everyone excited. So I think immune checkpoint inhibitors that interfere with certain immune regulatory mechanisms, CAR T cells, which are basically cells of the immune system hijacked to bind to a particular tumor and destroy it, antibody drug conjugates, I think, will be drugs of the future that hopefully, and we can really say only hopefully, transform cancer medicine.
Okay. And now talking about oncology in general, I want to ask you, what do you think is the most promising technology right now? And you cannot say ADCs.
No, I think it's really immuno-oncology in general. I think that was really the major paradigm shift in the last 10 years, that people have all of a sudden realized that you can only have a sustainable and successful cancer therapy if you really integrate with the immune system and the function of the immune system. If you simply try to kind of kill off cancer cells and you don't care about much else, you will always have the situation that some of those cancer cells will survive and then you have a tumor relapse and you don't have a long-lasting cure. Of course, there have been successes also in the past with standard chemotherapy, especially with combination with surgery, irradiation, etc., so standard therapies. But I think everyone gets so excited about the immunotherapies because it's almost logical that you have to use the immune system in order to fight disease. I mean, for antibiotics, it's clear, right? For many years, antibiotics won't clear your infection. It just helps to gain the immune system time to get rid of the infective agent. So if you consider cancer like an infectious disease, I mean, it's kind of course, a cell of your own body, but it has gone out of control. And in such a way, you can consider it a pathogen. But you need to engage the immune system for an effective therapy. And I think, in that sense, I cannot say this molecule is better or that cellular therapy is better. I think all cancer therapies involving the immune system will be successful eventually if, of course, the immune system is engaged in the right fashion.
Okay. So when you say eventually, I guess it's because there are still several problems with some of these therapies that are so new.
Yeah, that is true. I mean, let's say the biggest successes, of course, come with tumors which are so-called hot tumors or immunoresponsive tumors, which naturally have a tendency to respond to immunotherapies. But there are certain tumor types that have been resistant to these types of tumor therapies, so-called cold tumors, where the immune oncologists kind of scratch their heads, what do they have to do in order to make those tumors responsive to all of these innovative therapies? And I think it's currently the holy grail to find ways how to make cold tumors responsive to those new tools that the oncologists have at hand, which, by the way, are very much have entered into clinical development, and not all of them, of course, have already entered into the market. So we are still in early days. But I think, I mean, there are a large fraction of tumors which do not yet respond to these therapies, and we have to find ways and figure out what can we do to increase the chance of success for those difficult to treat tumors?
Okay.
I mean, yeah.
And you think ADCs can help with that?
I think ADCs, especially if they encompass the two principles that we have, the tumor targeting plus the activation of the immune system, as we have it in our molecules, I think that has a high chance of getting success. But we should not forget, as you said at the beginning, with antibody drug conjugates, the safety is one big hurdle, and the safety of these molecules is somehow determined by the design of the molecules. As I said, weak linkers, non-specific conjugation, you can quickly run into troubles, and this easy principle will then not be successful.
Okay. And so we've been commenting on a lot of different technologies that are in oncology, so it seems like there's a lot of competition, a lot of different companies, different technologies. Do you think this is a problem for the field, or is it a problem for you as a company to try to compete with all these other companies?
Well, it's always a problem if you have competition, but I think competition makes you stronger because you have to consider what your competitors are doing and where other people see promise in therapies, therapeutic principles. I think in that dialogue and in this kind of Darwinian system, I mean, survival of the fittest, if you wish, of course, the system by itself will select out the best therapeutic principles. And I think everyone in each company has to work as hard as possible to be selected. And sometimes one has to be lucky also to find the right combination of molecule, technology platform, and then things have to fall into place. There's a lot of research involved. I mean, it's not that everything can be planned, and one has to do a lot of work in testing to kind of make decisions to go for this or the other strategy.
All right. Then I want to ask you now also about a bit entrepreneurship, because so you founded MBE Therapeutics, but before MBE you founded Antibody, and you successfully developed the company and sold it. So I wanted to ask a bit about your experience as a founder, and especially founding a biotech company, which is obviously different than founding any other type of company.
Well, I think it's fun.
Okay.
I mean, first of all, I can say whoever thinks about founding a company or starting a company, I think first and foremost it's really fun, because I mean, you can really follow your ideas and you can follow your dreams. Well, at some point of time, of course, these dreams have to become some marketable reality, because if it stays a dream, I think it will, at the end, not commercially be successful. So, but it's a unique chance that you have to kind of discuss with your founders team or with your stakeholders what kind of strategy you would like to implement. So at the beginning you have an idea, then you may have, as Stefan said, a business plan, a business concept. I mean, then you pressure test that with, friends, with people from your network, and all of a sudden it becomes, well, it develops a dynamic when things then all of a sudden you see, well, this could really fly, this could be realized. Although, I have to say, this is not a kind of straight line. I mean, sometimes you see in business courses, I mean, these are kind of the growth curve of a company, and it's always linear or exponential. I think it goes, what I always say, in kind of waves. So you have these, kind of peaks of high hope, and then you have the setbacks, and then I call it the humps of iteration, when things go backwards and forwards until you find your way. But I think it's fascinating, because you're not getting into a fixed system, into a set system, but you really develop your own environment. You are kind of the master of your own disaster, if you wish, which I find fascinating. And I've never regretted to actually switch from academic research to more applied research, and really, well, developing something that hopefully could end up at some point of time in the clinic.
Okay, but do you still find it fascinating when you are at the low?
No, then not. Of course, if you are in a setback phase, or it kind of goes downhill, and I mean, there are always phases where things get difficult and tough, you have to have some persistence. I think persistence, everyone knows, right? I mean, you have to be persistent everywhere, every time in life. But there are phases, and they are inevitable, where things get a little rough, and then you have to kind of hold the faith and stay in there. I mean, not of course if you see there is no way forward. I mean, at some point of time you have to realize to maybe go around a certain obstacle. But generally speaking, I think the joy has an overweight against kind of the frustration that occasionally you do have. But who doesn't have frustration in a normal job, right? I mean, sometimes you have that as well. I think it's a normal fact of life that things go in waves.
Okay. Yeah, I mean, it's also, I think all entrepreneurs always tell you, failure is also a part of success. And
I think it is. I think indeed it is. I think one learns more from failure than from successes, because if you only have successes, well, you don't realize what could have gone wrong, and how you could have reacted in certain situations and reflected more about the situation. So I think there's always a positive, even in a negative.
Okay. So why don't you tell us about a failure at your previous company, from which you learned the most? You can share one.
You didn't tell me about that question. Well, I don't really have concrete examples, but of course, let's put it this way. I think it's almost generic. I mean, if in an innovative, let's say, startup company, you always face financial pressure, right? I mean, the saying is cash is king. And initially you don't have revenues unless you are a service business, but if you develop something, it's a lot of investment until finally you can realize revenues or even profit. So you're not profitable, you're burning money, which means inevitably that you come into situations where you don't have enough money at some point of time, or where you risk to not have enough money to continue. So this is where things get really, difficult, and one should not end up in a situation, and maybe this has happened a little bit sometimes in the past, where it gets too close to the cash out. So it's always better to have enough reserve to be more comfortable in the negotiations. But I mean, it can go very quickly because I mean, this is a capital-intensive business. It's not like IT where you have a computer and maybe a cell phone and you can do some web programming. You have to have a lab infrastructure. You have to have many people. So therefore, I mean, these initiatives like Base Launch are so fantastic because initially, I mean, where do you get the first money to get infrastructure or access to infrastructure? So money is always key. You can run low and one should not run too low too late.
Okay. And so also both of the companies we founded were based here in Basel. So I want to ask you, like, what are the advantages you see about, specifically being based here?
Well, first of all, I mean, academically, I have my roots here in Basel. So I did research at the Basel Institute for Immunology, which, for those of you who are a little bit older, know was a Roche-financed institute, similar to the Friedrich Miescher Institute from Novartis. So this is where I did my PhD, where I did research. And then when the institute was closed, it was not really a decision almost that I took myself. All of a sudden, I was in a situation where I thought, what can I do with my life? And then I thought, well, either go to university or maybe start a company. So yeah, it was kind of almost serendipity that I started my first company, built this one, I'm very proud that I built this kind of to a certain size that it was attractive for another company to be acquired. And in fact, the first molecules that were developed in that company are now in the clinic. We haven't reached that with the second company yet, but we are getting there.
Okay. And do you see any disadvantages to being based in Basel?
Oh, yeah, that was your question. No, no, I think I only see advantages, to be quite honest. And I'm not paid by Base Launch and not by Basel area. No, but I can tell you that with the first company, we had a subsidiary in Germany because originally I'm from the big canton. I have the feeling that the support by the authorities, by decision makers locally in politics and economics, I mean, there's much more support and active support here in Basel area than I experienced kind of in my own home country. And in particular, there's a certain level of, the people are very practical. If you have a problem here, everyone tries to help you and makes practical suggestions, even authorities. I shouldn't maybe bash too much my home country, but there it's very formal. It's sometimes very formal. You have to fill out 20 forms, and what is one PDF document that you send actually to the authorities here is at least one big binder with 3 printed copies for 20 forms that you have to submit to the authorities in that country, but maybe that's not in Berlin, but at least where I had the subsidiary. So it was more complicated. So I have the feeling on one hand, there's more support for entrepreneurs. It's a very entrepreneur-friendly kind of environment. There is, of course, especially in biotech and pharma, this is the place to be. Also because we have with Roche and Novartis 2 big institutions that create a big network here. So I think it's a very good, it's one hotbed of biotechnology in Europe, that area here.
So not a single disadvantage?
No, I don't think so.
Okay. All right.
Otherwise I get beaten. No, but I think it's really, it's very good. Yeah.
Yes, fine. Let's just let the audience, does anyone have any questions that you would like to ask us?
No, it will be exclusively injected because those are large antibody protein molecules, so they would be digested in the digestive tract, so they will not survive the stomach. So they are typically injectables that are intravenously injected. Even with the toxin attached to the antibody, a subcutaneous application would probably be too dangerous because then you have a high local concentration, so it will be intravenously injected, either in an infusion solution or really as a bolus injection. Yes, that is actually one very attractive approach that many companies are trying to achieve. We have tried that as well. To have a specific trigger, which maybe only exists in the tumor microenvironment, would be desirable. It's very difficult. So what we usually do is we implement certain generic cleavage sites in the linker that should only be activated once the ADC is bound to the cancer cell and internalized into the intracellular compartments. So there are certain proteases that are known to be active there, and then you can have certain recognition motives that then only release the toxin inside the cell. Having said that, of course, if that is released, they can also diffuse out of the cell in most cases, so you will always have some, I would say, collateral damage, even if you precisely control the kind of mechanism of cleavage. But it's a very good question. Well, I can tell you in my first company, I still worked for a long time in the lab still, because that was, and maybe that is one thing where I could say maybe that was a little bit of a failure, to be too long in the lab myself, trying to kind of, well, also implement or realize to practice the innovation. Because there are too many requirements, exactly as you say, in fundraising and writing business plans. And my first business plan, I mean, if I look at this today, it's a catastrophe. I hope nobody ever sees that and digs that out. But that was probably coming from the academic research a little bit counterproductive, to stay too long in the lab, to kind of, well, be part of that innovation, because that is where my heart was at the time, and still is, to be quite honest. I have to force myself out of the lab. I mean, of course, now I'm so detached from it that I will only cause mayhem in the lab if I enter. So my team is much better than I am. But yeah, it took quite a while, and this took actually quite some time also. Yeah, we do. Certainly we do have the vision. I mean, first and foremost, we have the vision that we want to develop drugs that are useful for patients. And of course, in order to achieve that, I mean, you have not only to go through research and preclinical development, but also clinical development, which is a tremendously expensive exercise. So it is clear that, of course, we had to take investors' money on board, and investors usually expect that they get a return on their investment. So that kind of takes a little bit of a limitation to your long-term vision. I mean, clearly as kind of our vision, or let's say midterm goals, as you said, is that the company will be so successful in developing their drugs that either we can take the company public, or that a big pharma company feels so much in need of getting access to the technologies or the asset, probably more to the asset, that we are developing clinically, that they will acquire us. It would be maybe with, I mean, taking investors on very, very early, from the research stage on, would be great if we can make it all the way to the market, but this will be a very long haul. I mean, companies like Actelion, of course, did that, but it took them 20 years as well, but with an enormous, of course, return on investment, if you wish. But very few investors have the patience to stay with a company for 20 years. They usually have an investment horizon of maximally 7 years. At 10 years, it gets really difficult. Wow, it gets really technical. Maybe I start first with but it's a very good question. Maybe I start first with a top-level view. It is indeed true, although the principle of ADC sounds very easy, and I've said at the beginning, it's actually quite difficult in realizing the concept, that there have been plenty of failures in the past with ADC molecules, mainly, in my opinion, due to the faulty design of the molecules. But that did have a profound impact on big pharma companies who actually, one after the other, have pulled out of this industry. I mean, I could name various pharma companies who said, well, they won't touch ADCs anymore because they had programs and they burned their fingers, mostly related to toxicity issues. It takes a whole lot of data and conviction to bring those players back to the table. In most cases, it's probably a futile exercise. But there are new players in the field who are a little bit more open and still believe in that concept. And interestingly, I have the feeling that there's more of an adoption of that concept in the Asian world. So the US companies and European countries, they have had a lot of negative experiences with these molecules. And I have the feeling they're slowly more divesting, and there are only maybe a handful of companies that are still active in the field. I'm talking about large pharma companies. But once we get successes from, let's say, Asian companies, be it in Japan or China or Korea, I bet that they will be very quickly back to the table and ask, well, if they can do it, why can't we do it? And we need to be in that principle as well. Because I'm firmly convinced if you do it right, and you combine the specific targeting with immunotherapy, which was what we discussed at the beginning, then you have a compelling principle. Well, I believe it's better if you internalize, because then, let's say, killing of the tumor cell comes from the inside. If you have non-internalizing ADCs where you rely on the deployment of the toxic payload by the extracellular matrix, of course you have less of a control. You only localize maybe in the tumor microenvironment, but there can be more diffusion happening, and of course tumors are vascularized. It goes into the into circulation. I would foresee more problems with that, but that's a personal opinion. There's no right and wrong. I don't know. Well, I think the homogeneity of the ADCs is also important, because then you have predictable PK properties which translate into predictable safety profiles. And I think that has also been a limitation of the conventional ADCs, that there's random heterogeneous conjugation and you have species in your drug substance which has unfavorable safety and PK properties. But that's a side note. Well, of course, in humans we don't know yet, because we haven't been in patients. This is where we want to go. And I ask exactly that question. All of the pharma companies who have actually clinical development programs, and they have always evaded an answer. They never said yes or no, they've always evaded it. What I can tell you that, of course, if you go with a humanized antibody, with whether you have a toxin or not, and you go into mice or monkeys, of course you raise immunogenicity to all parts of the molecule. We still have to make the experience whether what we see, let's say, in a foreign species with a human antibody will translate into humans. What I hear from, let's say, large pharma companies, and sometimes I have then the opportunity to talk to the speakers offline, they actually tell me that there are not much problems with anti-drug antibodies, and I don't believe it. I somehow don't believe it. I think there should be anti-drug antibodies. But the question is, how severe will it be? How often can you dose? What will happen there? And we will closely monitor that. Hopefully in 2 years I can answer that question from our own experience. Currently I can only speculate.
All right, time's up. If you have any remaining questions, you can just approach Wolf later at the networking session.
Thank you.
Wolf.
Thank you. Thanks.